Hi, we're now speaking to Professor Cavalieri about his recent work on social wasps and the yeast that mates inside them. So Professor Cavalieri, could you please give us a short summary of the work?
Yeah, so it's in my opinion, it's a nice example of interdisciplinary work. So we always think about interdisciplinary studies as something that mixes simply, different like physics and biology or big data and, mathematics. And this goes back to the classics. So this is an interaction between entomologists and microbiologists to try to answer a fundamental question on the ecology of Saccharomyces cerevisiae. So a few years ago, with the previous PNAS paper, we discovered that the intestine of the crabber wasps, is the big hornets, it can harbor Saccharomyces cerevisiae strains that are the same type and, let's say, same breed of those that perform wine fermentations. And we basically closed the cycle because we said, okay, the wasps have a buccal apparatus strong enough to break the berries of the vines, basically, and inoculate through trophallasis. So basically, bringing yeast that they have in the intestine, putting them in the berries, and then so the berries are loaded with yeast. And then when you crush the berries to make wine, fermentation naturally starts. And this happens not only during the months of September and October, but only throughout the winter. So we showed that the queen wasps hibernating can carry Saccharomyces cerevisiae. So the big question was, is this just, preserving existing variability? So this is relevant for the conservation and closing the cycle of Saccharomyces cerevisiae, or maybe this is a place where something more happens. And this something more, the big question, the big unanswered question was basically, where do yeasts have sex? Because we all know that yeasts have a cycle in which they produce an ascus, they're ascomycetes. The ascus is basically like a bag, and within this bag there are the 4 products of meiosis, so 4 spores. We know this very well, and we know extremely well the genes involved in this process for what happens in the lab. We had no clue so far and before these studies of where this was going to happen in nature. So the big question is, where is the place in nature where this process takes place? And we thought immediately of wasps. Why? Because this process requires time, because we know that sporulation starts when the yeast is starved for nitrogen. So you need to find a place where you have a little nitrogen, and intestine, the early part of the intestine, is a great place for this to happen. And then you would expect to give to this time to the cells to produce spores, and then for the spores to mate. We know that this is a long process. It's not a process that even in the lab, it takes at least a week or 2 weeks for sporulation. And then, the time that it takes for germination and mating is totally unknown, at least in nature. In the lab is a few days. So what we did, since we started looking at the microscope at dissected guts of the wasps, and we observed that within the guts we could see asci. So we could basically see these small bags with the 4 spores inside. And so the big question, are these asci forming within the gut? Are they taken from the environment and then transferred? What's going on? So instead of doing an observational study, we made a real genetics experiment in which we fed the wasps before hibernation with strains of Saccharomyces cerevisiae with different mutations, for which basically the only way to get viable crosses was if 2 different strains were going to cross one to the other. So we had genetic markers to be able to follow them. We also took Saccharomyces cerevisiae and Saccharomyces paradoxus, and we fed them to the wasps. So basically, this was like the wasp's gut was a big melting pot when we were looking at what was going to happen. And the other hypothesis we had is that this happens in social insects that hibernate, so that pass the winter overwintering, not like larvae, like Drosophila does. I mean, Drosophila passes the winter like larvae, like eggs. So it's basically there is not the entire animal, while the queens, the wasp queens, do hibernate as entire animals. And so to make this story short, then we sacrificed animals at different times during hibernation, and we plated and we diluted extremely well the colonies, and we basically checked with the genetic markers that we had in the strains what was going on. We discovered that, so 3 fundamental things. First of all, in the early days, so in the early samplings, you would not get hybrids at the beginning. After like 4 weeks, you would start seeing hybrids, but these were hybrids of Saccharomyces cerevisiae and Saccharomyces cerevisiae. Later on, the hybrids of Saccharomyces cerevisiae and the Saccharomyces paradoxus were the most abundant ones. So these somehow were showing, and the third thing that was also extremely important, Saccharomyces paradoxus, the second species, could not survive in the wasp's gut. So you would never find Paradoxus alive after a couple of weeks that it was spending within the wasp's gut. This was extremely interesting because Saccharomyces paradoxus is the wild counterpart of Saccharomyces cerevisiae. So it's basically discovered and found in bark of trees, while Saccharomyces cerevisiae usually is thought to come from vines, from the wine, from the winery, whatever, or from the wild also. And so these were 2 different environments and with 2 different species, and we could show that the place that where these good species mate and form viable hybrids is exactly the wasp's gut. Why this is relevant? We know since centuries that several of the strains that are important for the wine production or beer production are hybrids between Cerevisiae and Eubayanus, or Cerevisiae and Paradoxus. But we had no clue on where these hybrids were forming. We are sure that they do not form during beer fermentation or wine fermentation. So this discovery is extremely important in our mind because it actually shows a fundamental role of hibernating wasps and hibernation social insects in the ecology of Saccharomyces cerevisiae in the wilderness, so outside of human intervention. And since recent studies showed that the so-called domesticated Saccharomyces cerevisiae, I would say those strains that are more successful when we amplify Saccharomyces cerevisiae via wine fermentation, basically these strains have genes that are like, a little bit like a puzzle. So they are a mix of pieces of chromosome from different species. So these studies are bringing the evidence that what we have called Saccharomyces cerevisiae, the domesticated cerevisiae, the fittest for wine production or beer production, is basically, has a mixed genome with pieces from different species. Now what we are finding is the place where the genome get combined and scrambled. And we are finding that the place where the genomes are combined and scrambled is the gut of the wasps and probably of several other insects.
Wow, so I'm guessing this means like if something happens to the insects, we're not going to have wine and beer production happening. So should we be doing a lot more protective work? That sounds quite frightening.
This is an interesting thing because we usually give things for granted. Let's say winemakers, beer makers. I mean, you don't see yeasts. You don't see them. And so you give it for granted that they're there, that they exist. And a lot of people say, well, who cares? We can inoculate. So we can artificially reproduce the cycle by inoculating. And third, they say, well, after all these bugs, maybe do they really matter? Now, so we really understand is that yeasts matter for several aspects of the ecology, because probably yeasts, and we are working on this, have a fundamental role in maintaining the fitness of the insect. So there has to be a real mutual interaction. Second, insects like us, are holobionts. So they are the functions that we have and the insects have are the result of the symbiotic interaction between the animal and the fungi, or the animal of the bacteria. And destroying one of the two probably causes alterations in the ecosystem that we really cannot estimate. My take is that these alterations can be extremely dangerous, and I can tell you why. I mean, we are trying to do systematic studies, but one of the things that we are realizing is that in places where either by chemical warfare, because we use a lot of insecticides in the field, either with chemical warfare, not maybe in the vineyard, but in the apple orchards or somewhere else, anywhere you reduce the insects, we have well, so far statistically not significant, but we have observations indicating that you have a loss of biodiversity in the wine fermentations or in the cider fermentations, and there are more problems in getting them started. So we actually think that the insects are providing a fundamental part of ecosystem services that are useful for several aspects, and we should maintain them. There is another very important thing. Crabro wasps and insects like this serve in lotka volterra equilibrium against pests, because they are like the lions. It's like, what happened in Australia when they basically eliminated predators. I mean, you get rabbits, and the rabbits, are important and eliminate land. So predators are important, and in this case, the wasps are predators. And we are starting to think that maybe the way they feel Drosophila also uses some chemical signals that could be generated by fermentation, or so the same that yeast use to attract the wasps. So we think that this complex ecological system needs to be preserved in the same way where we preserve tigers or elephants in national parks in Africa. We think that we have to move to a new idea of conservation, bringing conservation to the next step, where we conserve the holobionts as part of the holosystem.
Wow, it's really interesting finding out just how important wasps are, because I think most people just dismiss them as being a bit annoying, but yeah, they're very, very useful. What do the wasps actually benefit from producing these yeasts? So I know we're getting a benefit, but are the wasps also getting a benefit?
So, this is a very interesting question, because we don't know yet. This is basically a very active field of investigation. But again, we are starting to have evidence that maybe the insects could provide an advantage for survival in extreme cold temperatures, because fungi and yeast produce secondary products of fermentation, like just glycerol, threalose, that are basically molecular chaperones, are like anti-freezing molecules, so that could be an effect. Then yeast provides supplements like vitamins, like vitamin B12, vitamin K, and so we think that hibernating wasps might get an advantage of being fed on yeast versus those that are not being fed on yeast. But we will tell you more with the results of the next study, because I mean these are experiments that are currently ongoing. But there are several known examples where fungi produce a fundamental service to the insects, and ants are one of these examples. Termites, and so very likely wasps probably have something similar than termites with respect to fungi like Saccharomyces cerevisiae.
That was really interesting. Thank you very much. Yeah, definitely going to look out for your next piece of research on this. We'll put the link to the paper. It's published in PNAS, and it's going to be put below this video. Thank you very much for joining us.
Thank you very much for the interview, and all the best with your channel.
Thank you.
Bye-bye. Have a nice day. Bye-bye.
Bye.